So students, we are just going to start with the new topic, that is testis. Yesterday only, or in the previous lecture only, we have just seen the testis is lying there inside inside the scrotal sac, which is completely safe inside the scrotal sac, and the purpose of testis to get descend down in the scrotal sac is to maintain the required temperature, whatever is required for the development or maturation of the sperm only. So now we are moving ahead, we are moving into the details of the testis only. So just look at the points, whatever I'm just going to discuss here. The testis is considered as the primary gonads in the human males. So in male reproductive system, the testis is considered as the primary gonad, and you knows very well what is the meaning of primary gonad. Primary gonad means what? The structure which is responsible for the release of gamete or formation of the gamete. So testis is the structure which is going to synthesize the gamete, means what? This is the structure which is going to form the spermatozoa and releases some of the hormones. Yesterday only, in the previous lecture, we have just discussed about the nature of the primary gonads. Primary gonads means the structures which are going to form the gamete sac cells and release the hormones. So here the testis is going to release the spermatozoa, to form the spermatozoa, and going to release the male sex hormone, that is called the testosterone. One very important point, yesterday only we have discussed the testis and ovary both are mesodermal in origin, and they both are being originated there in the abdominal cavity, and at the time of maturation, at the time of requirement, they are just moving downside into the abdominal cavity. So here the testis once again is of the mesodermal origin. I'm just going to revise once again. The testis is primary gonad in the human males, which is mesodermal in origin. And why this is primary gonad? Because this is responsible for the production of spermatozoa or sperm, and this is responsible for the release of testosterone. So student, now we are moving one by one in the detail of the testis. The first one, the location of the testis. Normally we knows very well, the testis originates in the abdominal cavity, and finally it is moves downside into the scrotal sac. Why this is so, you know very well. In the previous lecture only we have discussed the testis descend down into the scrotal sac to maintain the required temperature for the maturation of the sperm. So this is common. Testis will originates always there in the abdominal cavity and finally will be placed into the scrotal sac. And from where do it originates? In the embryonic development. During the embryonic development, it will develop from the mesodermal layer. So now we are moving on to that. This is not necessary in all the animals. The testis should always present there in the abdominal cavity. Are you getting my point? This is not necessary. The testis should always occupy the place there in the abdominal cavity. Based on the different positions, how many positions of the testis may be there? So just see, the first position, the testis permanently present in the scrotal sac and functions throughout the year. Are you getting my point? Permanently present in the scrotal sac and functions throughout the year means to say what? This is the case of the continuous breeder. Continuous breeder means what? The organisms, the animals, those who are having the breeding capacity to breed throughout the year, those animals are having their testis permanently located inside the scrotal sac, because they can continue to mature their spermatozoa. So such kind of the location or such kind of the testis which is present permanently into the scrotal sac is present there in the lion, bull, horse, and even there in the man. We are having what? Our testis is permanently located there in the scrotal sac. That is why we are the continuous breeder. We are not the seasonal breeder. Next type of the arrangement is there. The testis may remain there in the abdominal cavity, but it descends down into the scrotal sac during the particular time of the gamete maturation. Are you getting my point? In the first situation, what we have seen, the testis permanently remains there in the scrotal sac, and in the second situation, testis may live there in the abdominal cavity, but in the time of at the time of maturation of the gametes, it must descend down into the scrotal sac. Means what? When the reproduction time or when the reproductive period of the animal begin, that time testis descend down into the scrotal sac. Rest of the time it must remain there in the abdominal cavity. ये clear हो रहा है point? मीन्स व्हाट कि जहां पे ऐसी कंडीशन आ रही होगी, द एनिमल्स कैन नॉट ब्रीड थ्रू आउट द ईयर, सो दैट काइंड ऑफ द एनिमल्स आर कॉल्ड एज द सीजनल ब्रीडर. सो इन केस ऑफ सीजनल ब्रीडर, व्हाट विल हैपन? testis will come down into the scrotal sac only during the breeding season, and after completion of the breeding season, testis ascends back, move back to the abdominal cavity and get non-functional thereon. Is it clear to you all? पहला क्या था, हमेशा scrotal में रहेगा so that कि it could function throughout the year, and the organisms those who are having, they are the continuous breeder. दूसरा condition ये था कि scrotum will the testis will come into the scrotum only in the breeding season और बाद में वापस चला जाएगा in the abdominal cavity. Example अगर हम इसका देखते हैं तो bat, otter and llama. Basically these three orders of the class Mammalia, Insectivora, Tubulidentata and Rodentia, these three orders of the class Mammalia, they are having the seasonal breeder nature. The testis comes downside in the scrotal sac only during the breeding season, otherwise it will remain there into the abdominal cavity. Now third arrangement of the testis is there. Testis always remains in the abdominal cavity. पहले अरेंजमेंट में क्या देखा, टेस्टिस में कम्स, टेस्टिस रिमेन्स ऑलवेज इन स्क्रोटल सैक. दूसरा अरेंजमेंट हमने देखा कि टेस्टिस ब्रीडिंग सीजन में आ जाए स्क्रोटल सैक में और वापस चला जाए. इन द थर्ड अरेंजमेंट टेस्टिस में ऑलवेज रिमेन देयर इन द एब्डोमिनल कैविटी, टेस्टिस हमेशा एब्डोमिनल कैविटी में रहे. और ऐसा क्यों होता है, क्योंकि हम जानते हैं कि टेस्टिस इज डिसेंडिंग डाउन इनटू द स्क्रोटल सैक जस्ट टू इंश्योर द लेस बॉडी टेम्परेचर फॉर द मैच्योरेशन ऑफ द sperm. But here in those animals what happened, the body temperature is almost less or almost equal to the maturation for almost equal to the temperature that is needed for the maturation of the sperm. समझ में आ रहा है? इन एनिमल्स में बॉडी टेम्परेचर इतना कम होता है कि वो आसानी से स्पर्म को मैच्योर कर सकते हैं. तो दिस इज व्हाट दे डू नॉट हैव द स्क्रोटम. समझ में आया? टेस्टिस में हमेशा एब्डोमिनल कैविटी में रहेगा, कभी स्क्रोटम में नहीं आएगा, क्योंकि इनकी बॉडी का टेम्परेचर लगभग इतना होता है जहाँ पे ये स्पर्म का मैच्योरेशन कर सकते हैं. तो सच एनिमल्स विल नॉट हैव द स्क्रोटम इन देम. सो एलिफेंट्स आर द एग्जांपल, व्हेल्स, सील्स एंड द प्रोटोथेरियन. व्हेल्स एंड सील्स आर द एक्वेटिक मैमल्स. So this is clear to you all. In location, 3 location always present in scrotum. In humans, that is what the continuous breeder. In testis present in scrotum during the breeding season only, बाद में वापस चला जाए. That is in bat, otter, llama, Insectivora, Tubulidentata and Rodentia. Third one, testis always remains into the abdominal cavity. इनमें scrotal sac absent होता है. उसका कारण पूछा जाता है, तो you have to speak like that. The body temperature is low enough to mature the spermatozoa. So now moving on to the next point about the testis morphology, we are just going to begin. The shape of testis. The shape of testis is compressed, ellipsoid and oval. What is the shape of testis? It is compressed, ellipsoid and oval. The size of testis, we are just discussing the length, width and thickness of the testis. The length of testis is around 4 to 5 centimeter. The width of testis is 2 to 3 centimeter and thickness of the testis is 2 to 4 centimeter. What is length? 4 to 5 centimeter. Width is 2 to 3 centimeter. Thickness is 2 to 4 centimeter. The weight of testis is around 10.5 to 14 gram. So once again in the morphology, compressed structure है, ellipsoid है and oval in the shape is there. Size, length 4 to 5 centimeter, width 2 to 3 centimeter and thickness is around 2 to 4 centimeter and the weight is 10.5 to 14 gram. So is it clear about the morphology? Now we are moving on to the structure of the testis, internal structure of the testis. So in the structure only, when we are discussing the detailed structure of the testis, we are studying the LS of the testis. In the longitudinal section, how the testis appears. So first of all, from the peripheral region, if we are just going to observe the structure of testis, testis is covered externally by some of the protective covering. Those covering are called as coats. So testis is covered externally by the coats. So testis is covered externally by the 3 layers, means the testis is covered externally by the 3 coats, 3 sheaths, 3 tunics. The first tunic is called as tunica vaginalis propria testis. The first tunic or first covering is called as tunica vaginalis propria testis. Tunic means always the covering. Remember, tunic means what? The covering. Vaginalis propria testis. So tunica vaginalis propria testis is the outermost covering. Outermost covering. सबसे बाहर की तरफ मिलने वाली कवरिंग है, एंड दिस इज द सीरस लेयर, एंड यू नोज वेरी वेल इन द प्रीवियस लेक्चर्स वी हैव मेड इट क्लियर. Cirrus layer means it the layers or the cirrus layer will have a kind of potential space in between them, and that space will be provided with some of the liquids so that the collapse will can be prevented. So that is a kind of protective function of the tunica vaginalis propria testis. Are you getting my point? Cirrus layer is there, so cirrus cavity will be there, will have a potential fluid present over there that will prevent the collapse of the testis thereon. And this is a double layered structure. Double layered structure means to say this is made of two layer, and as usual you knows very well, if the double layered structure, so the outer layer is called as the parietal layer, and the inner layer of the tunica vaginalis propria testis is called as visceral layer. So outermost layer is the cirrus layer with a potential cavity, which is double layered structure. Outer layer of the vaginalis propria testis is called as parietal layer. The inner one is called as visceral layer. The next one, the middle one, is called as tunica albuginea. Once again, tunica albuginea. Tunica albuginea is the middle layer, which is comparatively the dense layer and made of the white fibrous coat. So this is middle layer, comparatively denser and made of white fibrous coat. Are you getting my point? This is white fibrous coat, dense layer, and middle arranged just below the tunica vaginalis propria testis. The third one, tunica vasculosa. Tunic vasculosa means what? Vascular layer. So tunica means layer or covering, vasculosa means vascular covering. So this is the innermost covering which is vascular in nature. Vascular in nature means what? This is made of the thin network of the blood capillaries. Are you getting my point? This is innermost layer, vascular in nature. Vascular in nature means to say always having the blood supply or richly supplied with the blood. So tunica vasculosa is a vascular layer, and it is made of a network of blood capillaries which lines the testicular lobules. What are the testicular lobules? We'll just see in forthcoming lecture. समझ में आ गया? तीन लेयर है. ट्यूनिका वेजाइनलिस प्रोपिया टेस्टिस सबसे बाहर की लेयर है, एल्ब्यूजिनिया बीच की लेयर है, कनेक्टिव टिश्यू की बनी होगी, फाइब्रस टिश्यूज की बनी होगी, स्ट्रेंथ देना है, और वेस्कुलोसा पूरी तरीके से वेस्कुलर लेयर है, दैट इज मेड ऑफ द ब्लड कैपिलरीज ओनली. Now we are moving on to the some more structure. So these three are the coats. These three are the layers by which the testis is protected. These three layer protection is there. These three coats are there: vaginalis propria testis, albuginia, and tunica vasculosa. We are moving on to the next structure of the testis, that is called as mediastinum testis. Mediastinum testis. What happened? The tunica albuginea start depressing inward toward the testicular cavities, toward the cavity of testis. The tunica albuginea is going to make a shallow depression inside it, depressed inside, just to make a groove inside, a depression inside. That depression of tunica albuginea toward the testicular cavity or toward the cavity of testis is called as what? Mediastinum testis. We'll see there in the diagram, and that will be that time you will get a clear idea about that. So mediastinum testis means what? It is a kind of inward depression of the tunica albuginea. Now what happened? That tunica albuginea continues. The tunica albuginea is a shallow depression. Now this continue in form of a longer depression or deeper depression. Due to that deeper depression, that depression appears like a septa or partition. Are you getting my point? So the mediastinum is now deepens inward to form the septular testis or septum inside the testis, that is called as septular testis. So what is the meaning of that one? The mediastinum testis continues inside in form of the septum. That septum are called as now septula septum. Testis के अंदर मिलने वाला septum, the septum present in the testis. So is it clear? The mediastinum testis, which has been started from the depression of the tunica albuginea, now it deepens inside, और उस depression के कारण, उस लगातार deep होने के कारण, that will form a kind of septa inside the testis cavity, testis cavity. So that septum is called as septular testis. The septular testis, this is very important question. Due to the arrangement of the septular testis, due to the formation of septular testis, we can understand the testis may get divided into many of the chambers due to the arrangement of the partition. It seems like that the testis is now having so many chambers present there. समझ में आ रहा है? Due to the arrangement of that septum, testis appears to be divided into the many of the chambers, many of the parts, and those parts are called as lobules, and the lobules are of the testis. That is why they are called as testicular lobules. Are you getting my point? Due to the septular testis, now I am just concluding the statement. Due to the septular testis, what happened? The testis appears to be divided into many of the lobes, and these lobes are called as testicular lobules. Is it clear to you all? Due to the septular testis, now testis is inside divided into many of the chambers or partition, and that chambers or partition are called as now as testicular lobules. Is it clear? Lobules present inside the testis, that is called as testicular lobule. Very important structure, and you will be shocked to understand in a single testis there are around 250 lobules are present. Are you getting my point? How many testicular lobules are present? Just cram it. In a single testis, 250 testicular lobules are present therein. So moving on to the next, what happens or what is present there inside the testicular lobules? क्यों ये इतना importance है? We are just going to see the testicular lobules. Inside the testicular lobule, each testicular lobule, or we can say that from each testicular lobule 1 to 3 supercoiled tubular structures are being originated. From each of the testicular lobules 1 to 3 supercoiled tubular structures are being originated. These structures are called as seminiferous tubules. Are you getting my point? From each of the lobules 1 to 3 supercoiled tubular structures are originated. That supercoiled tubular structures are called as seminiferous tubules. Means what? Each of the lobule is having 1 to 3 supercoiled tubule, and these tubules are called as seminiferous tubule. What is the meaning of that seminiferous? The semen bearing tubules. These tubules are the actual site of spermatogenesis. Are you getting my point? These tubules are actual sites of the spermatogenesis. So due to the arrangement of the seminiferous tubules inside the testis, there is a dense network of the tubular arrangement, a kind of complex network reticulum is being produced, and you will be shocked to understand, or you will be shocked to know कि each seminiferous tubule may have the length of around 30 to 70 centimeter. Each seminiferous tubule may have the length of around 30 to 70 centimeter. So now it is a kind of shocking effect. Suppose कि 250 lobules are there, and each one is having a single seminiferous tubule, then two single seminiferous tubule is having 30 centimeter in the length. So you can assume what will be the length of the seminiferous tubule in a single testis. Are you getting my point? That is a kind of amazing fact. So just remember, just take care of that. We are moving on to that. Just seminiferous tubule, why this is important, we'll discuss this structure later on, because this is what the structural and functional unit of the testis. Structural and functional unit of the testis means what? The entire testis is meant for this structure only. The entire testis is made of this structure only. And why it is functional unit? Because the testis has the major function to form the spermatozoa, so that process is spermatogenesis. So these tubules are the site of spermatogenesis. Here the spermatogenesis will be completed. Now inside the dense network of these seminiferous tubules, they are having supplied with the blood vessels also, because they are vascular. They are having nerve fibers also, the sensory in the natures. Means the testicular lobules are sensory in nature, they are vascular in nature, they are being supplied with the blood vessels, they are supplied with the nerve fibers, and some more specified cells are present in between the forest of these tubules. Are you getting my point? We can say the forest of tubules because it's a kind of dense network. Then too they are having some specified cells are being observed there in the dense forest of the tubule, and these cells are called as interstitial cells. These cells which are present there in between the seminiferous tubules in the testicular lobule, these cells are called as interstitial cells. Interstitial, you knows very well, the cells which are present in between the spaces are called as interstitial cells, and these cells were reported by the Leydig in 1850. The cells were reported first time by the Leydig in 1850. That is why these cells are called as interstitial cells of the Leydig. Are you getting my point? So testicular lobules are vascular in nature, so richly supplied with the blood. Sensory in nature, so nerve supply is also present there. Along with that, they are having some specified cells present over there called as interstitial cells, and these cells were reported by the Leydig in 1850. That is why the cells are also called as interstitial cells of the Leydig. So what is the importance of these Leydig cells? Why these cells are important, or why these cells are present here on? So these Leydig cells are actually endocrine in nature. These Leydig cells are endocrine in function, means these cells are responsible to release, or these cells are having responsibility to release and synthesize the male sex hormone that is called as androgen or testosterone. Androgen is the male sex hormone, which is the group of many of the steroid hormone, and major component of that androgen is testosterone. So now it is clear to you all how the testis is important, how the testis is considered as primary gonad, because in form of seminiferous tubule it is site of spermatogenesis. Along with that, it is having interstitial cells of Leydig which is going to release the male sex hormone that is called as testosterone. Okay, is it clear? Somewhere it has been reported that Leydig cells are having some specific appearance. Some crystals are being associated with the Leydig cell. Those crystals are called as crystals of Ranke. Are you getting my point? It has been reported that the Leydig cells are being loaded with some of the crystals. Those crystals are called as crystal of Ranke. So the crystal of Ranke, what are the function of the crystal of Ranke is not clear till now, so we can consider that they are of the unknown function. Okay, so if we are just going to observe the diagram of diagrammatically, we are just going to understand whatever we have explained till now. So just try to understand. I just use different color to express the different codes. So testis is covered externally by the 3 layers. So here 1, 2, that is the first 2 layers, that is called as tunica vaginalis propria testis. The second layer from this one. Now the third layer from the blue color. So here just understand the outer one layer, the outermost layer is called as tunica vaginalis propria testis. That is tunica vaginalis propria testis. That tunica vaginalis propria testis is having the outer layer is called as parietal layer. The inner layer is called as the visceral layer, and it is having a potential space present in between, that is normally called as the visceral cavity, normally called as the serous cavity, which will be supported or loaded with the serous fluid. So this is clear. The first layer, the outermost layer is tunica vaginalis propria testis, is a 2-layered structure. The outer layer is called as the parietal layer and the inner layer is called as the visceral layer. Just below this, the second layer is marked with the red color, that is called as tunica albuginea. Tunica albuginea is the fibrous layer providing strength, providing protection and color, providing strength and protection, that is middle layer, and just below the visceral layer of the tunica vaginalis propria testis. The third one I have just marked with that blue color here everywhere, that lines the complete testicular lobes. The complete testicular lobes are being lined with that. The complete testicular lobes along with the tunica albuginea. That inner one layer or the innermost layer is called as tunica vasculosa. This is the tunica vasculosa that I have marked here. The inner one layer, that highly vascular, made of the rich blood capillaries, is present inside the testicular lobes. The lines the complete testicular lobes and the tunica albuginea. Now just see the another structures. Here what happens in such case, you can observe the tunica albuginea start depressing inside. Here also everywhere you can observe the tunica albuginea start depressing inside. That shallow depression is called as what? Mediastinum testis. Are you getting my point? The tunica albuginea start depressing inside. Due to the shallow depression, another structure is being formed. That structure is called as mediastinum testis. Now this depression continues inside. From every mediastinum testis, the depression continues inside from every mediastinum testis. You can observe well everywhere. That depression that deepens inside, and that depression seems like a kind of partition therein. So that partition is called as what? Septula testis. Are you getting my point? The mediastinum testis continues in form of the septum inside the testicular cavities, and that partition is called as now septula testis. And very important thing, due to the arrangement of these septula testis, you can understand well the complete testis is found to be divided into many of the chambers. Are you getting my point? These chambers are called as lobules, and the lobules are present there inside the testis. That is why they are called as testicular lobules. So is it clear? Testicular lobules formed due to the partition of the testis. So how many testicular lobules are present there in a single testis? Around 250 testicular lobules are present in the single testis. So is it clear to you now? Now moving on to the next, that each testicular lobules give rise 1 to 3 supercoiled, that pink colored structure, 1 to 3 supercoiled structures there inside each and every testicular lobules are present. These highly supercoiled structure, tubular structure, supercoiled tubes are called as seminiferous tubules. These seminiferous tubules present in 1 to 3 in each the lobules, and these are the site of spermatogenesis. Along with that, I have just marked some specific cells with the blue color here. These blue color cells are appeared in form of a specific crystal, appeared in form of a specialized cell in the forest of, why I just told the concept as the forest of tubules, because you can identify this as a complex network, a meshed network of the tubule. So here in between that meshed network, a complex network, some specified cells are also present. Those specified cells are called as interstitial cells of Leydig. Are you getting my point? Interstitial in between these, so that is why interstitial, that was reported by Leydig in 1850. That is why it is called as interstitial cells of Leydig. These interstitial cells of Leydig are known to release out the male hormone. Are you getting my point? Now what happened? All the seminiferous tubules comes out from the testicular lobule and get collected in the lumen of the testis. This portion will be considered as the lumen. This portion of the testis will be considered as the lumen. This portion will be considered as the lumen. So in the lumen, the seminiferous tubules which comes out from the testicular lobules, many of the seminiferous tubules joins together to form the thin network of the tubules once again. So those thin network of the tubules are called as retitestis, like that. This one is retitestis, this retitestis. So many of the seminiferous tubules combines to form the thin network of the tubule, that is called as retitestis. Now the process of combination of these tubules will begin. So what happened? From seminiferous tubule, the combination of seminiferous tubule will lead to the formation of retitestis. Now the combination of retitestis will form what? Vasa efferentia. Due to the combination or union of the vasa efferentia, what will happen? The ductus epididymis will be produced, and finally due to the union of the ductus epididymis, what will happen? The vasa differentia will be produced. What will happen? The vasa differentia will be produced. So is it clear? What happened? First of all, the retitestis is produced due to the union of many of the seminiferous tubule. Now the retitestis unite to form the vasa efferentia. Now the ductus epididymis unites to form the vasa differentia. So is it clear to you all? Now vasa differentia will move out from the testis and will carry the sperm away from the testis. So if any question is there, basically there in the NEET examination, questions are there, they are just going to trace out the flow of the sperm passage, or how the sperm is flowing there inside the testis and out, how it is moving out of the body. So they are just going to ask the question to trace the passage, trace the pathway, how the sperm is going to flow thereon, and they are just missing the passage, they are missing some of the structure, rearranging. So you need to arrange those structures. So likewise, if you are having this diagram very much clear with you, so you will not have any kind of the problem. So how the seminiferous tubules are the structures where the sperm has been produced. These combines to form the retitestis, they combine to form the vasa efferentia, they combine to form the ductus epididymis, they combine to form the vasa differentia. Finally, they are just releasing the sperm into the urethra. So what is the flow of sperm? What is the passage of flow of sperm? Is the seminiferous tubule Reti testis, vasa efferentia, ductus epididymis, vasa differentia and urethra. So is it clear to you all now? Now once again I am just going to discuss the diagram LS of the testis. So listen once again. Here the outermost layer is called as tunica vaginalis propria testis. Tunica vaginalis propria testis is the outermost layer, that is serous in nature, means made of a double layered structure. The outer layer is called as parietal layer, the inner one is called as the visceral layer. Okay, now the middle one just below that is a fibrous layer called as tunica albuginum. The innermost layer is the vascular layer which lines the inner part of the tunica albuginea, and the complete testicular lobule it is going to line out. So that is called as tunica vasculosa. Tunic means the layer covering, and vasculosa vascular, and made of completely the blood supply or rich network of the blood capillaries. Next structure, the tunica albuginea depressed inside, start depressing inside, start invaginating inside and forms the structure that is called as mediastinum testis. In very next, this mediastinum testis continues inside in form of the septum. This mediastinum testis continues inside in form of the septum, and that is going to form the structure that is called as septula testis. Are you getting my point? The mediastinum testis continues inside the testis cavity in form of the septula testis. Due to the arrangement of septula testis, what happened? The testis is found to be divided into many of the cavities, and each cavities are called as lobules. The lobules are present in the testis, so that is why that lobules are called as testicular lobules. Is it clear? So each testis is having around 250 testicular lobules. Now you can observe in each of the testicular lobule there is a fine network of the tubules are present. So each of the testicular lobule give rise 1 to 3 supercoiled tubules. Those tubules are called as seminiferous tubules. In each of the testis 1 to 3 supercoiled tubules are there. So these tubules are the site of spermatogenesis. Here spermatogenesis process will complete. Next to that, in the dense network of the seminiferous tubule, some specified interstitial cells are present that has been reported by Leydig. That is why these cells are called as interstitial cells of the Leydig. Are you getting my point? And these leading cells are also very important since they are associated with the release of the male hormone, that is testosterone. Now what happened into the next, all the seminiferous tubule comes outside from the testicular lobules into the lumen of the testis. They joins together to form the reti testis. Now reti testis united together to form the vasa efferentia. Vasa efferentia forms the ductus epididymis. Now ductus epididymis combines to form the vasa differentia. So sometime the question is there regarding to asking the flow of the sperm passes. So flow of sperm, what is the pathway of sperm? Seminiferous tubule will synthesize the sperm, then it will be transferred into the reti testis, finally transferred into the efferentia, then to ductus epididymis, then to vasa differentia, finally urethra, and then the process of insemination occurs, the process of ejaculation occurs. The process of ejaculation occurs by which the semen, the sperm along with the semen will have to be discharged outside of the body. So this is all about the structure of the testis, so that we have completed today. So I think now everything is clear about the LS of the testis today. Everything is clear about the structure of the testis today. So major point, each and every point I have just cleared, and I tried to explain it in the simpler terms so that you will not have any kind of the problem. Just go through the video. Remember always, if you like the video, so please press the like button thereon. I will continuously trying to upload new videos related to your syllabus, related to cover the syllabus of the NEET also. So just try to go with those one and subscribe the channel. Okay. And in the next video I will just discuss the structure of this seminiferous tubule along with the other structures like vasa efferentia, ductus epididymis, means next portion of the human male reproductive system. So please like it, subscribe it. Thank you.